Thanks a lot for having me to give this talk. I'm really excited to get to tell you folks about it. It's not geographically related, it's biologically related. I got slotted in here at the last minute, so if you were here for something geographical, probably be a good time to step out. Or if you're interested in learning a little bit of biology, then feel free to join in. So yeah, Recursion Pharmaceuticals, I'll give you just a really quick background of myself and, there we go, that's better. Of myself and the company. I did electrical engineering, met this guy named Chris, started an internet company, went to grad school in molecular biology here at UT, and Recursion came from Chris's PhD. We started the company in March of last year. We now have 9 people, 6 are biologists, a few computational folks. We're actively looking for data scientists. That's one reason I came back to Austin. So here's what to expect in this talk. For about 3 minutes, I'm going to tell you a little bit about rare genetic diseases and biology. For about 6, how we find treatments for rare genetic diseases. And then 5 minutes, hopefully, I'm going to get to hop over into Jupyter for code and visuals. And I didn't get to prepare as good of a walkthrough as I hoped. This was kind of a last-minute talk invitation, but we'll see what we can do. So most people aren't really aware of rare genetic diseases. Most of them affect a very few people, each disease individually, but together they affect millions of people. People estimate about 10 million people just here in the US are affected by some kind of rare genetic disease. But they're really difficult to treat because they're small patient populations individually, so pharmaceutical companies generally haven't been motivated to make treatments because it's a difficult, expensive process. So this is one particular example of a rare genetic disease, and this one isn't super rare. There's somewhere between 50 and 100,000 symptomatic patients in the US. It is a genetic disease that you're born with, but it doesn't really show up until you're an adult, and you get these increasing numbers of these black spots that show up in MRIs on that brain on the left. This is called cerebral cavernous malformation, and you end up having a high likelihood for stroke, and there's currently no treatment available at all. Just to kind of whet your appetite a little bit, using the approach that I'm going to describe, Chris, during his PhD, was able to find a treatment for this particular genetic disease, kind of prioritize just a handful of drugs out of several thousand, and in this case, he tried 2 drugs on mouse models of the disease, and the pictures over there at the bottom left show that these mice, these are mouse models of this disease, they have all these pink expansions of the blood vessels in the brain, and both of these drugs were quite effective at reducing those numbers. So this approach that we're using, we have a hypothesis that this approach can work across many, many genetic diseases, and this is just the first one that we tried it on just to show you why we're interested in doing what I'm going to describe. So, and like I said, there are lots of these genetic diseases. So Chris, during his PhD, realized, hey, wait a second, what I just did worked for one genetic disease, but there's 2,000 that are basically in the same class of monogenic loss of function genetic diseases. Why don't I try to see if I might be able to do something that's much more impactful than even just finding these treatments for this one disease, which is great in and of itself. So how do we do that? So here's what Chris did, and these are pictures from his PhD. We have some healthy human cells on the left, and then there are ways that biologists have developed, and especially in the last 10 years really improved on, of messing up a particular gene that you care about, so you can model the disease, and that's the picture in the middle. And they sort of made an imaging-based approach for this disease to specifically highlight features of this disease, so they had a little bit of background knowledge for this disease, and that's why it looks so stark, the difference here. But it's the same principle as what we're trying to apply across diseases. And then, so once he had these human cells modeling the disease, he was able to use a microscope and take pictures and apply thousands of drugs to those disease model cells and try to find drugs that made the cells go back to looking healthy. And this is an exaggeration. What you're looking to find is identify the differences that seem to distinguish the disease from whatever you're considering to be healthy, and you're trying to find drugs that kind of act in a reverse direction to that. And this will illuminate a little bit more. So we're trying to take that approach that seems to have worked well for this one disease, while we're trying to move those treatments forward for that disease. At the same time, what we see as the bigger opportunity here is to try to, and what we've been doing for the last year, is taking that approach and basically making it more robust, that it could be applied across all of these 2,000 monogenic loss of function diseases, and I'll try to give you a sense of how that works. So we take pictures of the different cells, and up at the top you'll see pictures of healthy cells and cells from two different diseases. And then we use software that identifies the cells, segments them, segments a bunch of their internal components, and then just uses mathematical transformations to extract about 1,000 features from every cell that it identifies. So you're basically getting, for every single cell in these pictures, you're getting 1,000 measurements that sort of represent how does this cell look, and tries to quantify that as best it can. And so you get 1,000 features for every cell. What I'm showing down here on the bottom, the blue outlines didn't show up very well, did they? But I'm just trying to give you a sense of what we're looking for. This isn't what we do, but this is a subset of the 1,000 features that you'd see for each cell, and these are populations that have been averaged, where for each of these groups we have, you can see kind of 3 different rows for each group. And those are 3 different ways of trying to kind of model or attack the same disease. And what we're looking for is the things that those 3 ways have in common. And so disease A, you might be able to see the blue outlines where I've highlighted a few situations where disease A is a pretty strong standout phenotype. And for disease A, there are a number of those features where for all 3 different ways that we've tried to model the disease, the cells seem to be perturbed in the same way and significantly. And so that's kind of what we're looking for. And we tried to do that more systematically than if we had to look at all these heat maps, we'd go insane and it wouldn't scale to 2,000 diseases. So what we do is we represent each of these diseases. So every cell is its own point in this 1,000-dimensional feature space. And what we're trying to find are, to what extent for each disease are the cells kind of clustered together in this space. And we don't do clustering to do that. What we do is this is sort of a visual way of representing what we do for that. So you can imagine you have all these cells. We have tens of millions of cells in this feature space. A subset of them, maybe a few tens of thousands, correspond to different ways that we've tried to model this one particular disease. And one simple way to approach that, and we've done a lot of different ways here, but this is one simple way to talk about it, is to just kind of average all the points together for each disease and see where that lies in the feature space. And if we do that, what we're representing on the right is the 6 different colored blocks there represent the averages for, before I said 3, now I'm saying 6 different ways that we're using of modeling the same disease. And again, we want to find diseases for which those different ways of modeling line up so that we know we're seeing something that we can rely on and that we can move forward with. And so to answer that question, what we do, well, first I'll say this is just a nice way of visualizing that data that's 1,000 dimensions in 2 dimensions, where we find that average vector for all of the cells modeling that disease in this 1,000-dimensional space, and we just say, okay, let's put everything that's a projection along that vector on the x-axis, everything that's a distance to that vector on the y-axis. And now we have a visualization that shows us basically for this specific disease, it's called diamond black fan anemia. So now we have a specific visualization that simplifies the space down for us to look at it and try to make sense of what we're doing, where we have the diamond black fan anemia specific disease phenotype, the disease vector, the disease signature on the x-axis, and everything else on the y-axis. And that everything else is noise. These are noisy experiments. These cells do all kinds of things besides what we tell them to do. And then, like I said, we're using several different ways of addressing each disease, because each time we try to mess up the gene associated with the disease, it does a bunch of other things that we don't know about and don't want. So we're trying to kind of hone in on what those things have in common. And so that's visually what's represented here with this plot. Sorry, I lost my time. Come on, buddy. There we go. And yeah, so and how we decide whether we're doing well at that is we can basically train and test. And so we train on just 5 of these 6 ways of perturbing the cells. So we would look at just 5 of those blocks, and we would ask how close in the space. We measure by angles. We've tried a bunch of things, but how close in the space is the 6th? And you can do that. Just kind of leave one out, cross-validation, something like that, in a kind of angular proximity. way of thinking about it. And how much closer are all of those perturbations in this space than they would be if we just took a bunch of different perturbations at random that we didn't think were targeted at something in particular. So that's what allows us to build confidence around moving something forward so that we can try to find drugs. And this is one of those visualizations where we're actually trying to find drugs for one particular disease instead of modeling a bunch of different diseases here. So it's basically the same thing as the plot before. And over on the right are cells from the disease. Over on the left are cells from a bunch of other diseases. And we kind of think of the average of everything else as the healthy center that we're aiming for when we apply drugs, so that we don't find drugs that rescue something that's an experimental artifact. And so what we're looking for and what we see is we're looking for drugs, and we've put the drugs in red here, and the different sizes of the drugs are just the consistency between replicates. And so drugs that are moving directionally to the left from the green to the blue are what we want to see. And so we see one drug kind of down on the bottom left that seems to be doing something useful. And I don't have so I'm going to switch over into an IPython notebook in a second and show you how we can actually explore this data and not just have it there sitting statically. But that red dot that's farthest to the left might be something interesting for follow-up. And then the only thing I'll show you before hopping over into the notebook is this is just a way that we look at, like I said, how close these different angles were relative to a random shuffle. And so you can kind of make out this gray distribution that's between about 90 and 80 degrees there. And then the genes over, this is 50 different diseases. This is actually 60, that's a typo, sorry about that. But there's about 5 to 7 diseases over on the right side that once you kind of wrap your head around this data, you can say, okay, 5 out of 6 times or 6 out of 6 times, these guys are closer, way closer than you would expect by average. So we have something that we can be confident that's actually associated with the disease that we're looking at. And hopefully when we find drugs, we'll be finding something useful. So I'll quickly say kind of where we're going from here, and then I'll jump over into the IPython really quick. So right now, we're developing this approach on a pilot set of genes. We have about 110 genes right now, and we're pretty much finishing up with doing that so that we can scale these experiments across these 2,000 different diseases and hopefully find as many treatments as we can as quickly as possible. But there's a lot of analysis and a lot of optimization and decisions that are going into that. We're trying to develop better biological perturbations, so there's more of the effect that we want and less of what we don't. We are looking at, we've been playing with using deep convolutional nets to do better feature extraction than you can do with some of the existing tools out there and some of just these mathematical transformations. And then kind of right now, our business's strategy is basically testing as many combinations of drugs and diseases as we can, but we realize that we also, as we gather more of this, you can think of it as just kind of drug-disease interaction data. The more of it we gather, the more we might not need to test every possible one of the 1,000 diseases against every possible of the more than thousands of drugs. We might be able to use many different kinds of machine learning approaches to predict which ones might be more likely to work and prioritize our experiments better that way rather than kind of doing this exhaustive search of the space. So just have 1.5 minutes. Yeah, let's see. So all that I really wanted to show was that, yeah, we've been able to do this really quickly, and we love using Jupyter notebooks for all of this. So whenever an experiment happens, we can, this is kind of hard to make out on your screen there, but this is pulling every, so this was an experiment with 4 different plates of cells, and there's about 400 wells in every plate. And we can see, you can't even see from here, but each of those wells, there's 400 on each of these plates, has thousands of cells in it. But a biologist or even us computational folks, after looking at these experiments for a while, can get a sense of, okay, this experiment went pretty well. Actually, plate number 3 out of these 4 is kind of in bad shape. And so we can kind of notice things that we should incorporate into our QC process. And also just during analysis, trying to lift information out of this, we can say, okay, maybe we should just cut plate 3 out of this. This is just some of the ways that we've adapted and streamlined our own usage of bokeh, which is awesome. And so we do these interactive heat maps all over the place where we have, in this case, this is just representing, again, these 4 plates. And for these 4 plates, again, we can see, okay, plate 1 looks good, plate 2 looks good, plate 3, this blue everywhere means we have fewer cells than we expected. Something happened to plate 3, it's kind of messed up, and a biologist can come and explore this, and just using their mouse can hover over this and look around and see the different ones, and this is showing the actual source image from that experiment. It's hard to make out for you guys. The actual image of the full well is on the top, and then the bottom version is a zoomed in version showing a bunch of cells. So this well in particular, the zoomed in one is only showing 2 cells. That is a messed up well. So that kind of stuff makes it a lot easier for us to explore and figure out what we're doing and iterate faster to develop these methods and stay in constant loop, kind of a closed loop with our biologists, because we're developing all of these analyses directly in IPython, which we can directly share with the biologists, and they can interact with themselves. And it just makes it a really quick feedback loop. So I was hoping to get into more on the IPython side and show you some things about how we do the code, but I have to stop now. So thanks. Yeah. 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 So how we digitize the stuff. So yeah, we have a fancy automated microscope that takes these plates. These are 384 well plates that are about that big, and we grow cells on them, do a bunch of stuff to each well in the plate, and then we stick them in the microscope. A robot takes, we can stack 20 plates there. A robot takes them, puts them in the microscope. The microscope zooms along the plate, and in about 2 hours takes these high-resolution images of every well on the plate. So in terms of just capturing the images, is that what we do? Did you mean like segmentation, extracting the data? So for segmentation, for extracting the information from the images right now, we rely on there's a few different software tools that exist out there. The one that we like most and that we work with directly is Cell Profiler. And it's software that's been built specifically for this over a number of years so that biologists have kind of a WYSIWYG way of taking pictures of cells and designing different, just kind of old school thresholding techniques that help them segment the cells from whatever experiment they're in, and then it applies a bunch of mathematical transformations to extract information from each of those cells. Is this all visible light, or do you have fluorescence? Yeah, good question. There's fluorescence here. So yeah, we don't just do visible light. What we do is we use 6 different stains that attach to different types of cellular subcomponents and fluoresce at different wavelengths, and then we take pictures of those individually and then combine them at the end.
Thanks very much.
Thanks.